Central dogma is a core of molecular biology. This central dogma was proposed by Watson and Crick in 1953. So in this central dogma, we will see the information of genetic material will flow from one step to the other step, that is from DNA to RNA, then to protein. We can say that the information stored in the hard disk of the cell is transferred to the functional machinery, that is protein. So I have already told that the information is stored in the molecule of DNA, and this information is then transferred to a molecule of RNA, and RNA information that is stored in the RNA is decoded to form different types of proteins, and all the functions performed in the cell is performed by the proteins. So whether humans or plants or bacteria or fungi, they all have cells, and in the cells there is a molecule known as DNA. All the information of the cell is stored in this molecule. So whenever the cell has to divide into two cells or more cells, first this DNA molecule has to be divided into two molecules. That is the process of replication. So this is the process of replication in which DNA molecule is replicated into two molecules. So after this replication, each molecule is transferred to the newly formed cell. So from this information is stored in DNA is then transferred to RNA, and the process is known as transcription. So this is a very specific process in which DNA is transferred to RNA, or more precisely we can say that the information that is stored in the molecule of DNA is transferred to the molecule of mRNA, and this molecule of RNA is a complementary of the one strand, or we can say antisense strand of DNA. So this information stored in DNA is transferred to mRNA, and in the next step we can see the step of translation. The information that is stored in RNA is decoded through a process known as translation, in which different types of RNAs are also involved. Apart from mRNA, there are ribosomal RNA that forms ribosomes along with some proteins, and they form ribosomes, and these ribosomes are involved in the synthesis of proteins. So it is a very detailed process, and the proteins once formed, they performed a very specific functions. After we can see here a specific shape of the protein. Once the proteins are formed, they are folded in a very specific manner to achieve a very specific shape. Proteins which are not folded, they cannot perform their functions properly. So it is a very important step that protein must be folded. So we have seen here a very specific functions of in the central dogma, and basically it is a flow of information from one step to second to third through a process of transcription and translation. So this is the central dogma of the molecular biology. But recently there are some phenomena that do not obey this central dogma. For example, there are certain viruses that have been discovered, and most important is the HIV virus that causes AIDS in humans. This virus has RNA inside the virus, And this virus, because it has only RNA and do not have DNA, this virus then can make a copy of DNA by using a specific enzyme known as reverse transcriptase. So the important thing is that the central dogma is not being obeyed by this HIV virus, because in this step we can see the RNA is being transferred or being transcribed into cDNA. The DNA that will be formed is called cDNA, means complementary DNA. So we can see here the central dogma proceed in this direction, but this virus has transcription in this direction. So it is the reverse of central dogma. So we can see another example. One example is that of measles virus. Measles virus, and this virus also has a RNA, and this RNA is used to make a copy of RNA. So again we can see that in the central dogma RNA must be transcribed into protein, but in this case in measles virus RNA is being copied to form another molecule of RNA. There is another phenomena which do not obey the central dogma of molecular biology, is the discovery of ncRNAs, and these ncRNAs are called non-coding RNAs. Although the ribosomal and transfer RNAs are also non-coding RNAs, but apart from ribosomal and transfer RNAs, there are so many types of RNAs that falls in the category of ncRNAs. For example, SRP RNAs, long non-coding RNAs, siRNAs, miRNAs, and there is a long list, snRNAs, snRNAs. So they all belongs to non-coding RNAs. So these non-coding RNAs, they do not code for proteins. So it means that the information that flows from DNA to RNA, and if in this case RNA is non-coding RNAs, the information will not be transferred to form the proteins. So these are some of the cases in which they do not obey the central dogma. So we have seen that central dogma of molecular biology is just the flow of information from DNA to proteins through messenger RNA. And we have also seen some of the phenomena that do not obey the central dogma. For example, the HIV viruses and some measles viruses and the discovery of non-coding RNAs.